Welcome to Summerlands. I'm your host, Eva. When most people hear GLP-1 now, they think of Ozempic, Wegovy, Zepbound, weight loss, shortages, celebrity headlines, and one of the biggest pharmaceutical stories in the world. But that is not where this story starts. This story starts with a biological signal that, for a long time, looked almost too elegant to be useful. Scientists had identified a gut hormone that seemed to do nearly everything you'd want in the metabolic therapy. It helped stimulate insulin when glucose was high. It suppressed glucagon, which meant it could reduce the liver's tendency to release more sugar into the bloodstream. Food stayed in your stomach longer, plus it actually reduced appetite in the brain. And in principle, it looked like a remarkably intelligent system, targeting multiple pathways of metabolic dysfunction simultaneously. And the problem was that it disappeared almost as soon as it appeared. In the bloodstream, native GLP-1 lasted for about 90 seconds. And that, in a way, is the beginning of the whole story. Today's episode is about how this fragile, fleeting hormone over 4 decades so that the human body would actually accept it, then into a trillion-dollar category that escaped diabetes and began to reshape how medicine thinks about obesity, cardiovascular risk, metabolic disease, and maybe even prevention itself. And before GLP-1 became a culture phenomenon, it was a scientific and engineering problem. Before it became a blockbuster, it was a delivery problem. And before it became a delivery problem, it was a physiological mystery. So that is where I want to start. To understand why GLP-1 mattered, we have to look back to a much older question, one that had nothing to do with obesity and nothing to do with modern pharmaceutical competition. The question was simple. How does the body know when to prepare for sugar? And we need to go back 60 years ago. In 1964, a researcher named Neil McIntyre and his colleagues were trying to map out how the body manages glucose. They knew the pancreas produced insulin to clear sugar from the blood, but they didn't fully understand the trigger. They gave patients the same volume of glucose in 2 different ways. One group received the glucose intravenously, so injected straight into the bloodstream, and the other group received it orally. They just drank a glucose solution. If you think about the body in purely mechanical terms, you might expect the intravenous route to produce a stronger insulin response. The sugar is entering the blood immediately. It bypasses digestion. It should look like the more direct and more urgent metabolic event. Oral delivery, on the other hand, is delayed by the mechanical breakdown in the stomach and the absorption rate of the intestinal lining. It is a slower process. But that is not what happened. The insulin response was actually much larger when the glucose was taken orally. And that result forced a deeper realization. The body wasn't simply reacting to blood sugar after it appeared, it was anticipating it. The gut was not functioning as a passive tube that absorbed calories and passed them along. It was acting as an endocrine signaling system. As glucose passed through the digestive tract, the gut was sending an early message to the pancreas. Hey, a massive caloric load is currently being processed. Starting manufacturing and releasing insulin now before the bulk of this glucose even reaches systematic circulation. That phenomenon became known as the incretin effect. And it is not a minor biological footnote. In fact, it's responsible for up to 70% of the insulin secreted after a meal. And 70% of your body's insulin response is driven by these early signals from the digestive tract, rather than by glucose alone. In other words, the body doesn't wait for damage and then respond, it focuses. And once researchers understood that the gut was participating in glucose regulation that way, the next question was obvious. What exactly is the signal? Over time, scientists identified two major incretin hormones, GIP and GLP-1. For a while, both looked important, but the paths quickly diverged. GIP, although physiologically relevant, turned out to be much less useful as a therapeutic entry point in people with type 2 diabetes. In the diabetic state, the body's responsiveness to GIP was diminished. The signal was there, but the system was no longer listening very well. GLP was different, even in the context of metabolic dysfunction, where the body was highly resistant to insulin. GLP-1 still seemed to get through. And what made it so compelling was not just it stimulated insulin, it was the way it did it. GLP-1 enhanced insulin secretion in a glucose-dependent manner. That means the signal is conditional. It pushes the pancreas when glucose is elevated, but it backs off when glucose falls. That makes it fundamentally different from Belton therapies that can overshoot and drive patients into dangerous hyperglycemia. And GLP-1 was doing more than that. It also suppressed glucagon, which helps prevent the liver from releasing more glucose into circulation when it is not needed. It slowed gastric emptying, flattening the post-meal glucose spike. And it acts on the brain, especially on pathways involved in satiety and appetite regulation. It literally revives the brain's desire for food. So now the picture was becoming clear. This wasn't just a narrow diabetes hormone, it was a broader metabolic regulator. You have a single molecule that forces the pancreas to work more efficiently, stops the liver from sabotaging the bloodstream, slows down the mechanical processing of the stomach, and revives the brain's desire for food. It is the ultimate Swiss Army knife for metabolic disease. And that is why, even early on, GLP-1 looked so promising. It was not solving one downstream symptom. It was acting further upstream in a way that seemed unusually coherent. And yet, despite all of that promise, it was still unusable. Because of the 90-second paradox, the moment GLP-1 entered circulation, the body began breaking it down. The main reason was an enzyme called DPP-4. You can think about DPP-4 as a biochemical shutdown mechanism. Almost the second negative GLP-1 is secreted by the gut and enters circulation. DPP-4 identifies it, binds to it, and cleaves it. It snips off the first two amino acids of the appetite chain. And once those two amino acids are gone, the hormone is effectively deactivated. It is like a broken key. It can no longer fit into the GLP-1 receptor. A 90-second half-life while intravenous injection, or maybe in an hour and a half if administered subcutaneously into the fat tissue. From the standpoint of normal physiology, that makes sense. These are transient, male-related signals. They are not supposed to remain elevated forever. But from the standpoint of drug development, it was a disaster. If you were trying to build a real therapy, 90 seconds is not a usable half-life. You cannot treat a chronic disease with a signal that vanishes before it can sustain a clinical effect. Native GLP-1 was too fragile to become medicine. So by the late 1980s and early 1990s, researchers were in a strange position. They had identified what looked like a near-ideal biological mechanism. It improved insulin secretion without the same hypoglycemia risk. It reduced glucagon. It affects appetite. It seemed to coordinate metabolism rather than simply forcing it. but they had no durable way to deliver it. That is a paradox at the center of this story. The biology looked elegant. The pharmacology looked impossible. And this is where the story becomes more or less linear and much more interesting. Because the first real breakthrough didn't come from perfecting the human hormone itself. It came from outside human biology altogether. In 1992, a researcher named John M., working in a federal-funded VA lab in the Bronx, isolated a peptide from the venom of a clay monster, a venomous desert lizard native to the American Southwest and Mexico. At first glance, that seemed like a detour. You may ask, why was he even looking at Gila monsters in the first place? Well, Dr. N's laboratory was focused on discovering novel vas-active peptides, so hormones that affect blood vessels and blood pressure. And animal venom is not just a single toxic chemical. It is a complex, highly involved cocktail of bioactive peptides designed to paralyze, digest, or induce massive symptom shock in prey, and hypothesized that within these aggressive cocktails, there might be previously unidentified hormones that happen to interact with mammalian receptors. So he's fractionating the venom of the Gila monster. He's separating it into its individual chemical components, and he isolates a peptide that's called Xandem-4. And when they mapped the amino acid sequence of Xandem-4, the structural homology, the similarity to human GLP-1, was roughly 53%. And Xandem-4 mattered because it resembled GLP-1 closely enough to activate the same receptor. But it differed in a crucial way. It was much more resistant to degradation. The enzyme that native GLP-1 so quickly couldn't dismantle Xandem-4 in the same way. That difference changed the field. Because now the question was no longer theoretical. Researchers were no longer asking what the GLP-1 receptors was interesting biology. They had a proof of concept for the entire GLP-1 agnostic class. It validated the hypothesis that drugging this imprinting pathway could yield tangible, durable improvements in glycemic control for type 2 diabetes. That the problem was not the target, the problem was the architecture. Xanthine eventually became exenatide, marketed as Byetta. And when it was approved in 2005, it marked the first real commercial proof that the GLP-1 receptor agonist class could work in patients. That was a major milestone. But if you look back at Byetta from the vantage point of today, we have to acknowledge the reality of exenatide as a clinical product. It was not a silver bullet. The half-life was extended, sure, but it still required patients to inject themselves twice a day. The side effects, especially nausea and GI discomfort, were significant. The pharmacokinetics were much better than native GLP-1, but still not smooth enough, convenient enough, or scalable enough to define the future of metabolic medicine. So, exenatide did something very important, but very specific. It proved the concept. It didn't yet build the platform. And that is where the story shifts from discovery into engineering. Because once the field understood that GLP-1 receptor agonism really did work, there were, broadly speaking, two ways forward. One path was to deal with the degradation problem indirectly. If the body was destroying GLP-1 too quickly through DPP-4, then maybe you simply inhibit the enzyme and allow the body's own native GLP-1 to persist for longer. That path gave us DPP-4 inhibitors. Mechanistically, the idea made sense, but clinically, the results were modest. Those drugs improved glycemic control, yes, but they didn't create the kind of durable system-level metabolic effect that would later define the GLP-1 revolution. They didn't produce major weight loss. They didn't escape diabetes as a category. So the second path, the harder path, wasn't one that mattered more in the long run. The path that don't just protect the body's own GLP-1, rebuild it. Take the human hormone as your foundation and then engineer a version that can survive longer enough to function like a real medicine. That is the path Novo Nordisk pursued. And what makes Novo a role in this story so important is that the company was not just solving for efficacy, it was solving for architecture. It was asking a much bigger question. Not just can we drug the receptor, but can we build a molecule that is durable, tolerable, scalable, and repeatable enough to support an entire therapeutic franchise? Novo's insight was that the answer was not just to make GLP-1 less visible to DPP-4. The answer was to change how it moved through the body. And this is the 2019 albumin story published by Kunisum and Liao. Albumin is one of the most abundant proteins in human blood. Its primary job is to maintain osmotic pressure, but it also acts as a carrier. It transports various hormones, vitamins, and drugs throughout the body. It is this massive, bulky protein that circulates continuously for weeks before being cleared by the kidneys. It was a half-life about 19 days in humans. In strategic terms, albumin is a transport network already built by the body. What Novo realized was that if they could engineer a GLP-1 analog that could temporarily bind to albumin, they could let the body carry the drug for them. Not permanently, that wouldn't work. If the drug was permanently locked to albumin, it would never detach to reach its receptor and do its job, but reversibly enough to hide, travel, and then release small active amounts over time. That was the elegance of the idea. At any given point, 99% of the drug is hidden on the albumin basis, and 1% is free-floating, actively sending out GLP-1 receptors to trigger the metabolic response. And as that 1% is used up or degraded, more molecules detach from the albumin to replace them, maintaining a perfect flat continuous baseline of the. And instead of trying to buffalo stability by building an entire foreign molecule that designed a modified human GLP-1 analog that could hitch a ride on one of the body's own longest-lasting circulating proteins. That is what led to liraglutide. Liraglutide was the first major signal that Novo's architecture was working. By attaching a fatty acid chain to the peptide and designing it to bind reversibly to albumin, they extended the half-life enough to support once daily injection. That alone was a major advance over Bayada's twice-daily dosing. It improved adherence, flattened out the pharmacokinetic volatility, and made the category feel more realistic as a chronic therapy. But it wasn't the final answer. Because if daily dosing made the class palatable, weekly dosing could make it powerful. And that is where semaglutide enters the story. Semaglutide matters not just because it became Ozempic and Wegovy. It matters because it was the moment the platform matured. The chemistry here is intricate, but the strategic point is simple. Novo kept iterating until it found a molecular configuration that was stable enough, potent enough, and long-lasting enough to sustain once weekly dosing while still preserving the biological intelligence that made GLP-1 compelling in the first place. and it didn't arrive there in one step. One of the details I like in this story, it is the internal designation. Semaglutide as compound 217. That number matters because it reminds you what pharmaceutical success usually looks like from the inside, not genius in one flash. Iteration, failure, refinement, repetition. Hundreds of attempts, most of which didn't work. Compound 217 means 216 didn't get there first. They either bound too tightly to the albumin and never activate the receptor, or they bound too loosely and were rapidly degraded by the enzymes. It is the delicate balance. The breakthrough for compound 217 was the utilization of a C18 desert chain. This specific 18-carbon configuration achieved the perfect thermodynamic balance. It extended the half-life in the human body to approximately 165 hours. That is what allowed for one weekly injection that provided smooth, continuous receptor activation. And by the time Novo reached semaglutide, the company was no longer just building a better diabetes injection. It was building something that could travel much further. Because once you have a molecule that can be dosed weekly, that is potent enough to drive meaningful weight loss, and that sits on top of a mechanism affecting not just glucose, but appetite. Gut-structured emptying, satiety, and broader metabolic signaling. You are no longer simply competing in a diabetes market. You are creating the possibility of a category escape. But before that escape actually happens, one more engineering barrier had to be challenged. And that barrier was oral delivery. Because even if weekly injection is a major commercial and clinical advance, it still assumes the future of this class will remain bounded by peptide manufacturing and injectable administration. And that is a real limitation. Some patients will never want injections. Some health systems will always prefer oral options when efficacy is close enough. And strategically, once you think in platform terms, the question becomes unavoidable. Can this move beyond injection at all? And that is where oral semaglutide matters. We're talking about the development of Rybelsus, which was approved in 2019. Scientifically, it shouldn't have been easy. Peptides are not natural candidates for oral delivery. They're large, fragile, and exactly the kind of molecules the human digestive tract evolved to destroy. Swallow a peptide and the stomach acid denatures it. Digestive enzymes break it apart, and very little survives in active form long enough to reach the bloodstream intact. So from a drug delivery standpoint, oral GLP-1 look almost contradictory. Novo's answer was not to change the whole biology again. It was to build a formulation chip sophisticated enough to protect semaglutide just long enough to cross the barrier. That is where SNAC comes in, the absorption enhancer used in oral semaglutide. What SNAC does, in practical terms, is create a temporary microenvironment in the stomach that is less hostile to the peptide, while also helping the molecule pass through the stomach lining and into circulation. It acts as both an acid neutralizing shield and a chemical escort across the biology border. It is not elegant in the same way that albumin binding is elegant. It is more likely a clever delivery hack. The efficacy is low. Less than 1% of semaglutide in the pill actually makes it into the blood. But because they can pack the pill with a massive dose of the real peptide, that 1% is enough to achieve clinical efficacy comparable to the injections. And strategically, it matters because it proves something much bigger than our diabetes product. It proved that Novo was not just defending one molecule, it was extending a platform across delivery formats. Daily injection, weekly injection, oral formulation. At that point, this was no longer a single product story. It was beginning to look like a full-stack metabolic franchise. And that is where company strategy becomes just as important as pharmacology. One of the easiest mistakes to make intelligent GLP-1 story is to jump straight from Ozempic to obesity and assume the category exploded overnight. It didn't. Novo spent years laying the groundwork for that expansion. And one of the most important steps in that process was Saxenda. Saxenda tends to get overshadowed now because compared with Wegovy or Zepbound, the efficacy looks modest. But strategically, Saxenda was one of the most important products in the entire category. It took liraglutide, the daily GLP-1 analog, and repositioned it for obesity. Not diabetes, obesity. It was approved exclusively for chronic weight management. And that is the true inflection point. Because before Saxenda, GLP-1 was still understood mainly as a diabetes mechanism. Useful, important, commercially relevant, but still anchored to glycemic control. Saxenda helped break that frame. It established the precedent with the FDA that a GLP-1 receptor agonist could be safely and effectively deployed in an overweight population without underlying diabetes. It also helped normalize the idea that obesity was not just a behavior issue or cosmetic issue, but a pharmacologically addressable disease state. Saxenda itself was not the product that changed public consciousness. Saxenda was only generating around 5 to 8% body weight loss, which was clinically meaningful but not transformative. The dosing burden was still there. It required a daily injection. And in many ways, it was a bridge. Bridge matters. Saxenda helped establish the clinical logic, the physician familiarity, and regulator precedent, the commercial infrastructure that would later make Wegovy possible. And when Wegovy revived, the category changed. This is a point where GLP-1 stopped being an important therapeutic class and became something much bigger. Semaglutide at the obesity dose, in a once-weekly injectable form, produced weight loss results that crossed a threshold. Not just statistically significant, not just better than older drugs, a threshold where doctors, patients, investors, and the public all began to understand that this was different. Because for decades, anti-obesity pharmacotherapy had been disappointing at best and dangerous at worst. The field carried a lot of baggage, stimulants, poor tolerability, weak efficacy, short-lived enthusiasm, and the persistent idea that weight loss drugs were somehow medically unserious. Wegovy helped shatter that perception. Once you start seeing roughly 15% of body weight reduction in your drug trial, the conversation changes. That is no longer a cosmetic trick. That starts to touch sleeper apnea, joint load, prediabetes, liver fat, blood pressure, and a broader set of downstream metabolic consequences. And once that threshold is crossed, obesity itself starts to look different. Not as a lifestyle issue staying outside real medicine, but as a central metabolic node connected to a wider range of chronic disease burdens. That is what Novo had effectively built by the early 2020s. Not just a diabetes franchise, but the beginnings of a much bigger metabolic category. And then Eli Lilly changed the benchmark. This is where the competitive story becomes especially important, because Lilly realized that competing molecule to molecule on pure GLP-1 is a losing battle. To disrupt a monopoly, you don't build a slightly better version of the same drug. You change the biological benchmark entirely. It chose a different escalation path. Instead of optimizing a single agonistic architecture around GLP-1 alone, Lilly moved toward multi-receptor agonism. This was the genius of tirzepatide, branded as Mounjaro, which was approved in 2022. Early in the GLP-1 story, GIP had been identified as an anchor incretin hormone, but one that looked much less useful in type 2 diabetes. The diabetic system seemed relatively unresponsive to it, so GLP-1 became the main therapeutic path, and GIP receded into the background. Lilly brought it back. The logic was not simply that GIP had suddenly become powerful on its own. GLP-1 and GIP have distinctly different distributions of receptors throughout the entire body. When GLP-1 heavily targets the brain to reduce appetite, GIP receptors are highly concentrated on white adipose tissue. It was the combining GIP with GLP-1 might create a different metabolic effect than either signal alone. And that is exactly what tirzepatide began to show. The details of the mechanism are still being refined, but strategically, the outcome was clear. Lilly raised the efficacy bar. The efficacy literally reset the expectation for the entire pharmaceutical industry. We were no longer talking about a 5 or 10% weight reduction like with Saxenda. We are seeing 20% or more of the total body weight loss. Efficacy that began to rival the physical outcomes of the invasive bariatric surgery, delivered entirely via a synthetic peptide. It was unheard of, and this sheer potency is what fundamentally changed the clinical denominator of the field. This arms race continually forced the underlying science to evolve faster for the benefit of the patient. Novo had built the architecture that made the category possible, while Lilly reformed what the top end of the efficacy could look like. That shift matters because once a category starts producing not just meaningful, but extraordinary metabolic effects, expectations around the entire field change with it. And the question is no longer whether GLP-1 based therapies work. The question becomes how far this class can go, how much weight loss is possible, how broad the downstream benefits really are, and whether obesity itself should now be treated as one of the central organizing problems in chronic disease medicine. That's why the competition between Novo and Lilly is not just a corporate rivalry, it is a scientific escalation. Novo's strategy was to solve the delivery problem, stabilize the architecture, and build the category patiently over time. Lilly's strategy was to say, now that the category exists, we can raise the benchmark and force the entire field to move upward. And that is exactly what happened. Because once semaglutide and tirzepatide delivered the level of efficacy they did, the clinical meaning of the class started to shift again. Weight loss was no longer the whole story. It was becoming a proxy for something broader. The possibility that metabolic disease, cardiovascular risk, liver disease, sleep apnea, and perhaps other conditions were all more tightly connected than the solid structure of the medicine had just suggested. That is where the obesity trials mattered. For years, obesity medicine existed in an awkward place. Clinically important, socially loaded, and scientifically underpowered. The field had a long history of disappointment. Many of the older therapies either produced modest weight loss, unacceptable side effects, or both. And that history shaped how the medical establishment throughout the category. And this is the part what made the new GLP-1 data so consequential. The numbers didn't just look better than the past. They looked different enough to force a reclassification of the whole field. With semaglutide, and then even more with tirzepatide, you began seeing levels of weight loss that crossed into a range where the downstream consequences could no longer be dismissed as cosmetic. At that level, you're not talking about a mild improvement in appearance. You're talking about the possibility of changing the course of disease. That is what made STEP and SURMON so important. They were not just positive studies. They reset the denominator. They changed what patients expected. They changed what clinicians thought was possible. And they changed what investors, companies, and eventually health systems understood the market to be. But even then, one question remained. What was fundamentally still a weight loss story? Or was it becoming something more medically essential than that? That is where SELECT trial in 2023 and the consequent FDA label expansion in 2024 changed the field. If STEP and SURMON made the class impossible to ignore, SELECT changed the way the class would be interpreted. The SELECT trial enrolled over 17,000 patients who were overweight or obese and who had pre-existing cardiovascular disease. Crucially, none of these patients had diabetes. SELECT was not asking whether patients could lose weight. By that point, we already know they could. It was asking something much more consequential. Does treating obesity with semaglutide actually reduce cardiovascular events in people without diabetes? That is a very different question. The patients receiving Wegovy saw a 20% reduction in major adverse cardiovascular events compared to the placebo group. And the curves of the two groups, those on the drug versus those on placebo, began to separate almost immediately after the trial started. The data heavily suggested direct systematic anti-inflammatory effects. The GLP-1 receptor are actually present on the endothelial cells that line the blood vessels. So the FDA looked at that data in 2024 and officially expanded the label for Wegovy to include cardiovascular risk reduction. This is the moment it crossed the Rubicon. It shifted the frame from lifestyle and body size to heart attacks, strokes, and mortality risk. It moves the drug class out of the cultural noise and into the center of preventive medicine. At that point, semaglutide was no longer just a drug that happened to help with weight. It had become evidence that obesity itself was not merely adjacent to cardiovascular disease. It was part of the disease architecture. That is a deeper classification here. GLP-1 didn't just produce better outcomes inside an existing category. It began to redraw the boundaries between categories that medicine had treated separately for decades. In 2024, Lilly's dual agonist Zepbound was approved for severe obstructive sleep apnea. By aggressively reducing the fat deposits from the airway and potentially outlawing the neutral respiratory drive. The drug resolved the sleep apnea in a significant portion of patients. And then in 2025, Wegovy is approved for MASH. That is the metabolic dysfunction associated steatohepatitis, which is a severe progressive liver disease driven by the fat accumulation and inflammations that eventually leads to cirrhosis and the need for a liver transplant. Historically, there were virtually no effective pharmacological treatments for MASH. But because GLP-1 reduces systematic lipid spillover and directly dampens inflammation, it literally halts the fibrosis in the liver. I'm talking about a single molecule treating the pancreas, the heart, the airway, and the liver. GLP-1 became a pharmaceutical platform for metabolic disease. Well, drug class can move from diabetes into obesity, then into cardiovascular risk reduction, and then into liver disease, and then into additional pipeline expansion beyond that. You're not looking at a one product success story. You're looking at a architectural shift in the medicine. And that is part of why the commercial response was so explosive. Demand didn't surge because one company market well or because one brand went viral. Demand surged because the clinical implication of the class became larger than the infrastructure built to support it. I have the annual reports from Novo and Lilly here. By 2025, Novo Nordisk's obesity care division generated 82.3 billion Danish kroner. Ozempic alone brought in over 120 billion. On the Lilly side, in just fourth quarter of 2025, Mounjaro generated $7.4 billion and Zepbound hit $4.2 billion. But the financial numbers honestly hide the operational crisis, because the demand was so violent that it completely shadows the global installed manufacturing capacity for peptides. The underlying friction here is that synthesizing a complex peptide like semaglutide or tirzepatide is not like pressing out the aspirin tablets. It requires solely phase peptide synthesis, incredibly complex purification process using high-performance liquid chromatography and massive, highly specialized bioreactors. Furthermore, these are sterile injectables. They require sterile fill-finish facilities to put the liquid into the glass syringe pens without a single microscopic contaminant. You literally had two of the largest pharmaceutical companies pausing their advertising campaigns and restricting starter doses because there are simply not enough glass pens and not enough raw peptide to supply the millions of patients. And they are the victims of their own biological success. Which brings us to 2026 frontier. Because the ultimate goal of the pharmaceutical engineering isn't just efficacy, it's scale. If the manufacturing of complex peptides is a bottleneck, the scientific imperative is to replace the peptide. And that is exactly what happened in April 2026. When Eli Lilly secured FDA approval for Findel, the generic name being orforglipron. Findel changes the physics of the entire market because it is a true oral small molecule. Why is a small molecule so profoundly different from an oral peptide like Rybelsus? Rybelsus was a brute force method of shoving that massive protein through the stomach wall. A small molecule, on the other hand, like a stim or ibuprofen, is a tiny chemically synthesized compound. It has a very low molecular weight. It doesn't require bioreactors or solid phase synthesis. It can be manufactured in massive vats using standard chemical processes. It is infinitely cheaper and faster to produce. But creating a small molecule to targeting the GLP-1 receptor was considered nearly impossible for long. The GLP-1 receptor is a class B protein-coupled receptor. Native GLP-1 or semaglutide is a large peptide that essentially acts as a giant plug that perfectly fills entirely carbon to activate it. The small molecule of a GLP-1 doesn't try to fill the entire carbon. Instead, it slips into a tiny, highly specific hydrophilic pocket deep inside the receptor. When it binds to that specific pocket, it physically forces the entire massive receptor structure to twist and change its shape, triggering the exact same intracellular signaling cascade as a giant peptide would. It is a mechanical hack. This innovation solves the manufacturing bottleneck entirely. You can press fungal oil into simple cheap pills by the billions, but while small molecules are solving the delivery and manufacturing scale, the peptide engineers haven't stopped pushing the biological efficacy. In 2026, we are also analyzing the phase 3 data for Lilly's next generation molecule, retatrutide. This is a triple agonist. It targets GLP-1, GIP, and the third receptor, glucagon. The data showed patients losing an astonishing 24% of their body weight in 48 weeks. We established earlier that one of the primary benefits of GLP-1 is that it suppresses glucagon. Glucagon is the hormone that tells the liver to dump sugar into the blood. If you activate the glucagon receptor, shouldn't you be causing a massive blood sugar spike and worsening diabetes? That is the paradox of glucagon biology. It is fundamentally a hormone of energy mobilization. When you activate the glucagon receptor in the context of extreme or caloric disaffected, it is being enforced by the GLP-1 GIP suppressing the appetite. The glucagon actually acts as a massive metabolic furnace. It signals the body to start actively burning stored fat for fuel at an accelerated rate. It specifically targets the clearance of fat from the liver. You have GLP-1, GIP, cutting off the calorie intake and sensitizing the fat cells, while glucagon simultaneously turns off the thermostatic to burn off the existing stores. And the GLP-1 prevents the blood sugar from spiking, keeping the glucagon's metabolic burn safely contained. It is the absolute pinnacle of physiological engineering. If we look back over the incredible journey we have charted for you today, in 60 years, science solved the biology of the incretin puzzle. Chemistry solved the impossible half-life problem, using lizard venom as a bridge and then engineering human albumin binding and the SNAC Trojan horse to protect the fragile hormone. And then Novo Nordisk and Eli Lilly solved the commercial strategy and the biological architecture, pushing from single targets to dual agonists to triple agonists to small molecules. They systematically isolated a biological whisper, amplified it, stabilized it, and engineered it into a role that can treat diabetes, reverse obesity, protect the heart, and help the liver. Many of the chronic conditions we have treated as separate silos may actually be downstream expressions of the same metabolic disarray. It is a triumph of scientific persistence. But there's always a but in medicine. The science works, the clinical evidence is undeniable, the commercial scale is staggering. We have engineered out our way out of the biological problem. We have built the perfect molecules, but that brings us to the system problem. Who gets coverage? Who gets excluded? Who pays for chronic treatment at that scale? How do employers think about a drug that may reduce long-term risk but adds major short-term cost? What do insurers do with a class that is medically important, financially disruptive, and societally visible all at once? That is where the real conflict begins. Because GLP-1 solved a biological problems first. Now it has collided with an economic one. And that economic problem may be even harder. Which perfectly sets up our next episode. We're going to wade into those access walls. We are going to look at the immense friction between the cost of these miracles and the reality of health plans. We'll look at the strategies payers are using to manage the financial of widespread GLP-1 adoption. Because with GLP-1, invention was only the beginning. Thank you for joining me today. See you next time.